actually. Okay. So, good morning, everyone, and welcome to the mini-symposium series of the European Society of Marine Biotechnology. So I'm going to share briefly my screen just to share with you some points of today. And all right, just a quick, all right. And I am sharing. Everyone can see it?
Yes.
Yes.
Okay, so we are, as I mentioned, the European Society for Marine Biotechnology. Just some housekeeping. If you can, if you're not presenting or talking at this point, can you? I will ask you to have your microphones muted. Then we don't have any extra sounds coming through. And really, I'm very glad that we managed to pull this symposium here because it is a chance for early career researchers to talk about the work. So a little bit about the ESMB. This society was established in France in April 1995. And actually, the idea was to promote biotechnology and research within Europe. But of course, it expands much more across than Europe and create collaborations. Of course, ESMB promotes the exchange of scientific knowledge, but also training and collaborations and cooperation that would all eventually help the society and the environment. So we do promote placements that we will be advertising, and they keep on coming. So any early careers, that would be great if would like to share, if they would be interested for a placement. Our main activities are ESMB conferences and webinars, meeting and training through placements. And of course, you can have the access to the Journal of Marine Biotechnology. And we do get newsletters every quarter of the month of a year. This is our main media. So we have an Instagram, we have our main web page, where in the web page we will upload this talk after it's finished. And then of course you can join in our wider LinkedIn community. So please make note of this media if you're interested to follow any of those, or you want to check the presentation again. So with that, I would like to just break through a little bit our symposium for today. And in this case, we will have Professor, Professor Juan Luis Gomez Pincetti. Did I pronounce that correctly? Sorry, yes. And who will talk about the collection at the Spanish Bank of Algae as a tool to microalgae conservation and research development in the blue biotech sector. So at this point, I'm just gonna give a little, okay, first of all, we're gonna then have 4 PhD student talks. Jorge Garcia Marquez, Marte Jensen, Shaima Hassad Bey Makbul. I'm so sorry if I don't pronounce your names correctly. And Hafid Al Marouki. So, so without further ado, I would like to introduce our key speaker. So Professor Gomez Pinchetti is a scientific director and head of the biotechnology unit at the Spanish Bank of Algae, which is also known as BEA. He's a scientist in alga biotechnology and a member of the Institute of Oceanography and Global Change. His main research interests are related to the biotechnology, physiology, and cultivation of marine macro and microalgae. So he has a broad experience in laboratory and pilot-scale cultivation of marine algae cyanobacteria. And his research objectives include physiological and biochemical characterization, transformation, and applications of algal biomass. So very interesting and very, topics that they are very relevant nowadays. So I will give the floor to Professor Juan Luis Gomez Pinchetti. However, I will ask you to, we will have a question at the end, a question session. Sorry, I'm gonna stop sharing now. So Professor will, once he finishes, you can pose some questions.
Yeah.
And but you can also put some questions on the chat room and we will follow that up. If there is no time, it's okay, because at the very end we will open the floor for any questions or discussions that might take place. Okay, so I'm now stopping and then I'm giving the floor to Professor.
Okay, thank you. Good morning, everybody. And many thanks for the invitation to this mini symposium. I'm very happy to be here and to share our work with you. Can you see the presentation?
Yes, Hafid.
Okay, perfect. Yes. Thank you. Okay, well, when Roberto and Maria already invite me to share with you this short presentation, I thought that I was giving to you some ideas of what we are doing in our facility, which is called the Spanish Bank of Algae, where we have been working for 30 years now, trying to develop the activities, the sector of the algal biotechnology in the Canary Islands, because we think that we are in a very nice place, which has all the resources to go ahead with this kind of diversification of activities, and particularly in the research area. We are placed in the Canary Islands, in the island of Gran Canaria, in a scientific park, which is called the Marine Scientific Park of the University of Las Palmas de Gran Canaria, which is placed in the Muelle de Taliarte. And here you can see an aerial view of our facilities, which is, well, it's not a very big facility, but it's very nice. Very nice. You see, we have a building with the labs and all the equipment, and we have here a greenhouse facility for the development of intensive and cultivation of marine macroalgae and microalgae, right, with tanks, and well, we'll see some slides later. You know, during this period, we have been developing all this kind of activity related to projects. And nowadays, our main area of research, what we call our biotechnological vocation, is related, I will say it in two main points. The first one is the conservation of biodiversity through well, we have a very nice collection of microalgae and cyanobacteria, which I will describe later. And with all this genetic material, with all these genetic resources, we are doing a program for identification and development of biomarkers related with biochemistry, molecular and flow cytometry, molecular biology, and then the development of cultivation techniques, the characterization of all these kind of strains for the biotechnological point of view, just searching for bioactive compounds that we can use in biotechnological applications related with agriculture, bioremediation, bioactives, functional metabolites for food and nutrition health, metabolites for the cosmetic industry, or algae for animal feed and aquaculture. These are our main topics, and we are full aligned with the Sustainable Development Goals, and we try to develop all this kind of activity through research projects and services and agreements with different companies that are interested to work with us in these kind of things. Just from in the beginning, 30 years ago, we were very you know, starting with the potential of seaweeds cultivation in the Canary Islands. You know, we have a very high biodiversity, but the populations are very small. So we started to grow them in intensive tanks, in tanks of cultures, making a mix of what is aquaculture with the use of the biomass and the characterization of biomass. But after that, we started we came into the microalgae work, where there was not previous activity in the Canary Islands, so everything was free for initiating research on this area. So we start to make bioprospection through the Canary Islands, and started to do a small collection, mainly focused on cyanobacteria and some marine strains of microalgae. But the collection has been growing very, very fast because the potential is very high. And here you have some examples of the bioprospection we made. We like very much the marine environment, but we prospect also in freshwater and continental waters in the islands, and also the stream environments. We like very much to work in this kind of environments. We are very nice to find species that work very well in the laboratory, in the things we do in the laboratory. Here you have some examples, some sampling in salt marshes. For example, this is a very curious one. 10 years ago, we went to the underwater volcano in Del Hierro Island, and we sampling there when the volcano was in erupting, and we got a small collection of cells that were living there in high CO2 concentration and low pHs, which is very interesting for us. And for example, in the last year, we have got different events related with red tides, and then we also go there and try to check what strains, what the species are growing there, and we make the bioprospection and work with these samples in the laboratory. This has made that we nowadays have maintained a collection which is more than 2,000 strains of microalgae and cyanobacteria related with different taxonomical groups, and we keep them in, you know, they are unialgal clonal strains, and we have to keep them mostly in alive, which is a very hard work, but we also make experiments on cryopreservation and maintaining of the strains in different possibilities for the maintenance of the strains. So through all this biodiversity that is very new for our places, because this is an original collection, we make the characterization and see the possibilities that we have with all these new strains that we are discovering in our samples, and we make the second step, which is the characterization of the strains and the possibilities from for the biotechnological sector. In the last years, with the experience we have been acquiring, we have developed different projects to make networking with the Macaronesian region, which is the region related to the archipelago of Azores and Madeira and Cabo Verde, and some regions in the north of Africa. And we have made a network of collections. At these universities, they are starting new collection from the local places, which are, you know, we are relating networking activities, trying to develop the biotechnological sector of microalgae in the Macaronesian region, which is a very, very nice activity, and it's a very nice project. When we have been going through the collection and considering the possibilities we have with all this biological diversity, we have also been in contact and coming to the two main consortium of laboratories in Europe, you know, what we call the ERICs, and we are involved in the ERIC MIRI, which is the Microbial Resource Research Infrastructure, which is related with all the microorganism collection that we have in Europe, those that are interested to come into this network. And from this information we got from the different collections, we have made a you know, flow diagram of the activities every of us can develop in the facilities, and through the ERIC we are having access to a lot of results and data that we got from the collection, but also we are trying to develop a program for the characterization of possibilities for the mass cultivation of all these kind of strains, with the objective to develop biotechnological activity in Europe. And here you can see how the flow diagram flows and what are the possibilities, because MIRI as an ERIC, they have the TNA programs where you can have access to the facilities and the materials that we have already in our laboratories. So it's a very nice activity, and it's very interesting for these new projects and new research which is going out from your laboratories and from our laboratories also. We are also involved in the EMBRC ERIC, which is the Consortium for Marine Biological Resources in Europe, which is very much developed or focused also in research about marine biodiversity, and particularly, well, different fields of research, but also they consider the possibilities we have with the microalgae. So there is a roadmap plan for the work with microalgae in Europe, and you can see here how is the discovery pipeline, which is described in the consortium, where we are focusing most of these steps you can see here in the figure. We have the microalgae isolation, the growth and characterization of the growth from the different strains, the possibilities to isolate new compounds coming from these strains and genetic resources, and also the identification of bioactivity with the objective to develop possible industrial application of this biomass. Nowadays, we are into the Spanish node of MBRC, together with the people in the Basque Country and the Galicia. You know, with all this information that we get from the collection, we are very much aligned and involved in the development of the Nagoya Protocol, just to apply the principles which are developed in the protocol according to the use and the industrial use of the genetic resources. So we have all the protocols, and we apply for all the information we can get from our collection through the Nagoya Protocol, which will be very important for us in the near future. Is already in development, so we all should be aware of the establishment of the Nagoya Protocol. Okay. The second part of the story is that with all this genetic material that we have already in our collection, we make different projects at different levels, in locals, the Spanish government and the European Union. And I will show you some of the examples we make nowadays. In the laboratory scale, we are working now with into the project New Tech Aqua, which is a project related with the improvements and new techniques developments in the aquaculture sector in Europe. But curiously, we are the strange guys for the microalgae in the project, and they propose us to make a very interesting thing that was not very much studied before, which is to study a selection program and breeding program with microalgae through classical breeding. That means we are working into the project with different population of microalgae, particularly diatoms, some strains of diatoms that we are using for make crossings and try to get new progeny coming from different parents, and try to identify possibilities for improvement of growth of these cells and also the biochemical characterization. It's it's a very difficult task, but we are being successful on the development of the process, and we have got some new progeny histories coming from parent cells, and we are trying to characterize all these new breeds that we have got around the project. The next is that together with the work we are doing in the laboratory, we have also access to very small but very nice facility for the development of cultures. As I say at the beginning of the talk, we are in a very nice place with very good environmental conditions. We can work the whole year around because we have sunlight the whole year around, and we have nice temperatures, ranges, and this is very good to go ahead with this kind of projects that we can, because we can grow algae in our facility just the whole year round. That means that we have different photobioreactor scales and raceways at different scales also that permit that we can do this kind of culture experiments in outdoor conditions and to obtain biomass for characterization of the from the biotechnological point of view. Nowadays, we are now building a new small facility which is going to give us the possibility to work with photobioreactors under controlled conditions. This is a new growing chamber where we will have a set of photobioreactors under light and temperature and pH and everything with a small robotic system, trying to obtain and work with different strains under controlled conditions to obtain as much results as possible on the biomass. With all these kind of experiments, we develop a program to characterize the different groups of substances that can be interesting from the biotechnological point of view. We have the facilities to go ahead with the downstream processes, just harvesting the biomass and then process them through dry techniques and also extractions. And we have got a extracto deck and a collection of extracts that can give us a lot of information when we work with these kind of things. We work with different groups of metabolites that I will show you now, but this is an example of the different things that we can do in the laboratory. Other of the projects, which is an example of the activity we made, is the Savannah project, which is a European project which will finish at the end of this year, but has given us the possibility to work in a very nice research area, which is the bioremediation. In the project, we have been working at the lab scale, but also in a demo plant facility where we have selected marine strains and other groups have selected freshwater strains of microalgae and cyanobacteria with possibilities for the agriculture sector and aquaculture sectors, and try to identify these selected strains to this kind of application and grow them in a demo facility from 1 hectare to 5 hectares and see the possibility, the real possibilities by using wastewaters, different kind of wastewaters to grow the biomass and to make the bioremediation processes and to obtain different final products related with the strains we have been selected. Here you have the example how we have selected different strains from 2 collections in the consortium, and we have checked the plant biostimulant effect to promote higher plants growth regulation. And from this, we have been working on the cultivation at these demo plant facilities. We have developed a very interesting projects and programs to characterize all these kind of new strains that we have in the collection, and we check for the different groups of metabolites that can give activities or bioactivities in different fields of interest. You know, as I have tried to describe, we have a very high biodiversity of strains that can give us a lot of information related with the increasing potential of this kind of metabolites from the biotechnological point of view. So we work with the different groups of metabolites, including polyunsaturated fatty acids, chlorophyll. Pigments is a very nice field for us because we have different taxonomical groups which give us different profiles of pigments. We work with carotenoids and phycobiliproteins, but also with exopolysaccharides, which well, are a very interesting area of research nowadays, because the possibilities for these kind of molecules are very interesting, and you have a lot of application, including the prebiotics and possible the use of polysaccharides for probiotics also in this field. We work with proteins and peptides and mycosporins and amino acids, and particularly we are also involved or working with polyphenols coming from microalgae, which are very interesting. As you see in the slide, we have different figures and pictures showing what we make in the laboratory, the characterization of the growth, but also the potential of how we harvest the cells in the exponential phase or the stationary phase to get the metabolites we are interested in. Well, these are some of the examples we have been working in the last years with some colleagues from Tunisia and also Madeira, and we have been we have made the characterization of some exopolymeric substances coming from some microalgae, but particularly this thermotolerant chlorophyte Graeciella, which grows in very high temperatures and is able to excrete a lot of exopolymeric substances. And we have made the characterization at the lab scale, but we are also looking for applications as the development of a biodegradable film that can be useful for different applications in food protection, for example. We have been also characterizing some amino acids and some mycosporin-like amino acids as UV protectant, but also as source of antioxidants for possible cosmeceuticals application. And we make also all this characterization of the different strains we can grow in the laboratory to identify phenolic profiles or phenolic components, and also related with the antioxidant activity of these extracts that we can get in the laboratory and make and see the possibilities of all these kind of strains for biotechnological applications. I don't know if I have gone very fast. I have not controlled the time, but this is more or less in a short presentation what we make in our laboratory. So feel free to contact us, and I'm available. Thank you very much.
Thank you very much for this nice presentation. So my name is Jeanette Hammer-Anders, and I'm taking over from Maria. So
okay, thank you.
Thank you. That was a really nice overview and also impressive collection you have. So I will ask the participants, if you have questions, please either raise your hand or just unmute. Yes, Marta?
Hey, thank you for the nice talk. It was really, really interesting one. I wanted to ask about this, because you said you sampled from a volcano.
Yes.
Did you get like any, what can I say, taxonomic surprises, or was there anything? Yeah, it's very, very interesting. 10 years ago, I don't know if you remember, there was an underwater volcanic eruption in the island of El Hierro, here in the Canary Islands. And you know, we have the possibility to go in a oceanographic cruise
in our oceanographic campaign, just because we were interested to see if under these conditions we were able to discover or to identify microalgae that were growing there in a very extreme condition. That means very low pH and very high concentration of CO2. And we got there some samples that were already living in the column, in the seawater column above the volcano. And we have here a small collection which was obtained from these samples. You know, one of the things that we have this small collection, and the problem with this is that we cannot reproduce the real conditions where these cells were growing at that time in El Hierro, because it technically is very difficult, but it would be very, very nice if we can have this kind of possibility. Or when we go to, for example, we have been doing bioprospection in Las Azores, in the volcanic area of the Azores, you know, and you can get there samples where you can have very special cells that are growing in very extreme conditions. But it's very difficult to have this in the same condition in the laboratory. It would be very nice to go ahead with a special project, you know, trying to fix these conditions and try to keep these cells under these conditions. Anyway, we have these cells that we got from this volcano in the collection, and they are available. And maybe some project related with this will be very interesting.
Thank you very much. Are there any other questions? Well, I asked the next speaker to just get prepared to share screen, but I have done a question. Yeah, you think about scale up, and I know there has been a lot of discussion. How do you harvest in the large scale? What's the strategy?
Well, there are different strategies depending on the morphology of the cells. We have different equipments, but the most common equipment nowadays is the centrifugation. There are a special centrifuge to harvest the biomass from the different systems. We have a big one. It's not the normal laboratory centrifuge, although we have a centrifuge with that we can use with bottles, big bottles of 1 liter. But anyway, we have a special centrifuge for this harvesting. And for example, with cyanobacteria, which are filamentous, we can use filtration. And we have also a special equipment that allow us to have this filtration, and we can get the biomass from there. There are different techniques, and they are very, very advanced nowadays, and you can have many possibilities for this.
Yes, thank you. Yeah, it will be interesting to follow the collection and the bioprospecting performed on that. So thank you very much for sharing that with us. And I think I will thank you, and I will ask the next speaker to share the screen. It's I have to apologize in advance, and now is Jorge Garcia Marquez. He's going to share with us the advances in European aquaculture and diversification. So please.
Good morning, Janet. Can you see my whole presentation?
Yes, I can see it. Thank you.
Okay. Hello, everyone. My name is Jorge Garcia. I'm a PhD student at the University of Malaga, and I'm going to speak about my research related to the nutritional quality of wild and farm thick-lipped gray mallets. Okay, so first of all, why I have mentioned aquaculture diversification in my title. The reason is because only 5 species are responsible for the 95% of the total European production in 2019. Those species were salmon, trout, sea ram, sea basses, and carps. So there is a clear need for aquaculture diversification in Europe. And in this sense, FAO has recently advocated for diversification concerning the species by increasing the number of species cultivated, increasing the illness of the cultivated species, and also increasing the diversity within the species currently cultivated. And I will focus on this first point, increasing the number of species cultivated, in particular with thick-lipped gray mallets. Which is a very interesting candidate for aquaculture diversification because it is an omnivorous species in the early stages of development, to change to herbivorous with the age. It has a high osmoregulatory capacity, who allows to inhabit a broad range of salinities without affecting its growth rate. Also, recently have been reported that Chelolabrosus fry production was more efficient in green water than in clear water, suggesting and also demonstrating the possibility to control its production in captivity. So with that in mind, we aim with this study to investigate the differences in the proximate composition, the fatty acid profile, and the amino acid content between wild and cultivated mallets. And the relevance of our study was to try to understand and to increase the knowledge on the nutritional attributes of wild and cultivated mallets in order to help to improve the design of diets following the requirements. For that, we collected 10 mallets, which were fed for 3 months with a commercial diet from tilapia, because there is no specific aqua feed for mallets. These animals were fed in the facilities of the Marítimo Saporito Center in San Fernando in Spain. And also we caught 10 wild mallets from a salt marsh there in San Fernando. We kept them alive and we transported to the center facilities where all the animals were euthanized, and we took liver and muscle samples which were immediately frozen into liquid nitrogen and kept until minus 80 degrees until the subsequent analysis from the liver, which was the fatty acid composition, and the proximate composition, fatty acids and amino acid analysis from the muscle. The results from the muscle proximate compositions, well, while Chelolabrosus has high crude protein and low crude lipids than cultivated specimens, this high content of lipid in cultivated species cultivated specimens could be related to a variety of factors like the species, the physiological status, or even the reduced activity of these farm animals. And in this study, in our study, the farm fish were fed with a commercial aqua feed, which was rich in protein and lipids. And so it could seem that the lipid accumulation in these farm animals indicate that they use preferentially the dietary protein for energy, while the fat extracted from the feed was stored in the body. The liver fatty acids, the high levels that we see in the palmitic and also the oleic acids seems to be related to the commercial feeds. And this is something that we expected because it is well established by the literature that this the dietary fatty acid composition of the aqua feed is reflected on what we see on the tissues, on marine tissues. We will also see the same pattern in mussels fatty acid profile. From cultivated fat, from the cultivated mallets, they had high monounsaturated fatty acids and total 9 fatty acids than wild specimens. That was mainly due to the levels, the high level of palmitoleic and oleic acids, also related to the commercial aqua feeds. And from the wild animals, they have a higher content on essential fatty acids like arachidonic, EPA and DHA fatty acids, also the polyunsaturated and the total N3 fatty acids. From the muscle fatty acids, it is important. The cultivated animals, they had high saturated and monounsaturated fatty acids, while the wild Chelolabrosus had higher polyunsaturated fatty acids. Again, like I just mentioned, on liver, levels of palmitic, oleic, and linoleic acids in cultivated specimens were higher. This is what I mentioned. It is because of the high content in aqua feeds. Within the N3 polyunsaturated fatty acids, the wild content of EPA and DHA was 3 times higher than the culture fish, 60 versus 20%. And also, if we add the content of arachidonic acids, the wild mallets have more than 65% of those 3 essential fatty acids, which they play a key role in the fish physiology and metabolism. But why is that so different? Because they are more than 3 times higher in the wild than the cultivated animals. So what we suggest is that this difference came from the natural feeding of these specimens, because these animals were fed naturally in the salt marsh of San Fernando, which is based on benthic diatoms and epiphytic algae, which are rich in N3 fatty acids. Also, this is very important. The difference in fatty acids is important, particularly from the point of view of the human nutrition. First, the wild content of EPA plus DHA was 50% higher in the wild than the culture fish. If we export this data into grams of EPA plus DHA per 100 grams of fresh fillet, we have more than 1 grams in the wild versus 0.77 grams per 100 fillets. And this is in line with that was published in other fish species like salmon or trouts. Also, we have to keep in mind that EPA and DHA plays a determinant role in preventing many human diseases like obesity or brain disorders. And for that, the FAO and other organizations recommend a daily intake of EPA and DHA at least of 250 milligrams per day. And our results indicate that that cultivated and wild animals can be considered as a good source of these 2 essential fatty acids in the human diets. On the other hand, the cultivated species had lower EPA and DHA content than the reference species, and this could be due to different concentration of lipids in the feeds. The lipid content in salmonids, for example, in the figures, they can reach up to 30%, and while lipid content on the commercial feed that we provided to the farm mullets was 6%. So maybe the difference can come from this different concentration of lipids in the aquafeed. So the question is, how can we increase the EPA and DHA content of in culture mullets? So we suggest that we can increase the percentage of lipids in the aquafeeds, but also we can increase particularly the EPA and DHA content in aquafeeds by considering supplementation with microalgae due to its good fatty acid profile, as they naturally fit with this kind of algae. The mallet's amino acid composition, well, except for histidine in the wild specimens, both cultivated and wild specimens showed higher amino acid concentrations compared to the reference that the FAO, the WHO standard concentration regarding to the human amino acid requirements. So both specimens from cultivated and wild, they can be considered as a good source of food with high quality protein. And as take-home messages, first, I will keep you in mind that only 5 species, salmon, trout, sea bream, sea basses, and carps represent the 95% of the European aquaculture production. So there is a clear need for species diversification. We aim to increase the production total of mallets. In this particular case, the pig lip gray mallet, Cerulabrosus. And we found differences in fatty acid and amino acid profile between culture and wild animals. The nutritional superiority of wild fish was observed. Also, the lower percentage of N3 polyunsaturated fatty acids in cultivated fish may reduce its nutritional quality. So for the industry should aim towards an increase in a total N3 polyunsaturated content of culture mallets choosing the proper dietary lipid intake. Consider supplementation with microalgae, like for example, a blend of microalgae like Nanochloropsis gaditana or Schizocridium, due to its good fatty acid profiles. And to finish these presentations, I will say that these results that I have presented have been published in the journal Animals with this title that you are seeing there. And I will thank you, and I will wait for your questions.
Thank you very much for a really nice presentation, and also I think a very important study to increase the diversification. So I will ask if there are any questions. Please raise your hand. Let me see. So while we're waiting, if people are thinking about the question, so I think this is important, showing that actually taking the time to study other species or to really try to do something with the diversification. But have you considered something about which microalgae that you would, if you were thinking of adding it to the feed? Have you already discussed these kind of topics?
Yeah, we had an experiment with chlorella. We actually fed these mallets with chlorella for 90 days, and we saw a good growth. And also we are analyzing data from the microbiota and the gene expression, but the results were good. We are aiming to publish it right now.
Yeah. Oh, that's good news. Thank you. And I
thank you for your question, Janet.
Yeah. So Conchetta has a question.
Yes, thank you. Good morning, Janet, and thank you, George, and the previous speaker, Juan. Thank you for your contribution. I'm going to, okay. So, George, your study is very interesting. As a research group, we are following the same research activities, as there is a great interest in contribute to aquaculture diversification and focusing on non-target and traditional species. I feel that your study is the occasion also to look on specific metabolisms of fish species, as this species is a peculiar metabolism related to its habit. So the adaptation to different climatic and environmental situation could be the one explanation of its different metabolisms, not dependent only, I feel, on the diet, but also on the different environment. So it is possible that in a situation like an intensive culture, the standard energetic level is the occasion, as you know, that an higher production of more energetic fatty acid is promoted in respect to the other, where in a natural environment, possibly more variable situation and also different component of the natural food web can contribute to the different accumulation of fatty acids. I find it is very interesting topic, and you are lucky to have time as a PhD student to study this basic and pure research aspect. But of course, with the challenge to contribute to give information to the productive sector. So it is a very good study. So, and of course, we are also able to have an exchange of opinion as research, thanks to the network of the European Marine Biotechnology Society. Thank you.
Thank you, Conchetta, for your opinion. Thank you very much.
Yes. So in interest of time, I said thank you very much again for this nice and clear presentation. I think we should move on to the next presenter. It's Marte Jensen, a PhD fellow at UiT, the Arctic University of Tromsø. So please, Marte, can you share your screen?
Yes, I will see. Okay, let's see here. second. Do you see my screen now?
Yes, and if you put it in presenter mode.
Yes, I'm just also gonna start the video. All right, let's see if it happens. There. Okay, so you see the screen and no comments, right?
Yes.
Okay,
Looks
perfect.
good.
Great. Thank you guys for having me here today. So as Jeanette said, my name is Marte Jensen, and I'm a PhD fellow in the Marbio Research Group at UiT, the Arctic University of Norway. So we are working on marine bioprospecting. And today I will talk to you about marine fungi as a source for new bioactive compounds, more specifically on the story of Lulvortinon. So I will just start. Let's see. Okay, so what I want to highlight here is just that the focus on marine-derived fungi has increased during the last couple of years, or the last decades actually, but the group of fungi that are considered to be strictly marine, they're often called the obligate marine fungi or marine fungi sensu stricto. They are not very well studied, especially when it comes to their biosynthetic potential. So there are several reasons for this, but some include that they're difficult to isolate in the first place from the natural environment, and also that they are quite difficult to grow in the lab. So I will also show you an example of this when it comes to low growth rates. So the fungal order Lulvortiales is a strictly marine order of fungi, and it has been studied for its distribution in different marine habitats, but its biosynthetic potential, so the compound it's able to produce, has not been studied before, or at least not been published. All right, so that takes me to the main part of this talk. I'm gonna walk you through this article that we published in the beginning of October about Lulvortinon, a new dimeric naphthopyron from a marine fungus in the family Lulvortiaceae, with antibacterial activity against clinical methicillin-resistant Staphylococcus aureus isolates. So this was a collaboration article at the UiT, the Arctic University of Norway. So the whole thing started with an isolate called 067BN1.2. So Associate Professor Teppo Rama here at the university isolated this fungus from a dead pine in the Pinus species in the splash zone in Kongsfjord, Berlevåg, Norway. So you can see the red dot there all the way up in the north of Norway. The fungus was taxonomically placed within the Lulvortiaceae family, which is in this order that I just mentioned, the Lulvortiales, which is strictly marine. So in 2017, when I started my PhD, we picked it out of the -80 freezer and put it onto agar. And when the agar was covered with mycelium, we used this as inoculum for liquid cultures. So just to give you guys some time perspective here, the time that I needed on agar for it to grow, to kind of cover the whole agar with mycelium, was about 40 days. And then we transferred it over to liquid, and in liquid, to produce good amounts of biomass, it needed to be inoculated or incubated for more than 100 days. So in total, it was incubating for more than 140 days before extraction. So as you can understand, it takes quite some time to cultivate this. Then the culture was extracted, and the extract was fractionated into 6 fractions using flash chromatography. The fractions were screened for bioactivity, mainly against or for antibacterial activity and for inhibition of biofilm formation. And we discovered interesting bioactivity in one of the fractions. So flash fraction 5 was then chosen further for dereplication and isolation. So very briefly, dereplication. This is the process of identifying potential interesting compound candidates in a fraction or in a sample. So we want to point out some potential candidates, and we also want to establish if these are new compounds or if they are known compounds. So what you see here is actually the first time I discovered or I saw lulwortinone in our HPLC high resolution MS. So usually this is a line, you can see the baseline and you have a lot of peaks, but in this case we got this huge mountain eluting over several minutes. This was then the compound that we have been studying. And as you can see, it was a really clear candidate for the activity in this fraction. So based on the MS data and also supported by the UV data, it appeared to be the by far major component in this active fraction. We established the elemental composition, as you can see here, to be C37H40O14S. And this did not give any hits in the relevant natural product databases. So because of the interesting bioactivity and also the fact that it seems to be new and also that there's a lot of it, we chose to go further to isolate the compound. So as we normally do to isolate compounds, we use this preparative HPLC MS. So what we saw also here was that the compound eluted over several minutes, as you saw in the previous chromatogram from our high resolution MS. So this was also the fact that it was strongly sticking to the columns, which made it a really inefficient method. Actually, I had to clean columns for several days to get rid of the compound. So we chose or decided that this was not the way to isolate this compound. And because there was so high contents of the compound in the fraction, we rather chose to isolate it using flash chromatography. And that worked out nicely. So what we got from the isolated compound was a green compound, which was for a long time named the green one, but now has gotten a more proper name, which is lulwortinone. So it's named after its producer. The structure of the compound was established by the NMR group here at the university by Johan Isaksson and Philip Rainsford, and they found that this was a dimeric naftopyrone. So as you can see here, I hope you see my little laser thing. It's a dimer, two monomers connected together here, and also substituted with a sulfate group. What they also observed was that the compound changed in the presence of acid. More specifically, they saw that the sulfate group migrated from the 6th position to the 9th position in the presence of acid. So this is yet another reason why we should not use the PrEP system, because this, in this case, we need to use acidified solvents, and we don't use that when we isolate with the flash. Yes, what they also observed from the NMR experiments was that the compound 1, the lulwortinone, seemed to aggregate with itself to form higher structures. And this model here, made by Johan, is showing you how the sulfate group could be facilitating this aggregation. So from this, we decided to continue with the lulwortinone for bioactivity testing. So it was tested in a variety of assays, and here I'm going to show you the ones where it was active. So for inhibition of biofilm formation, we test this against S. epidermidis, and I will show you here the results. On the y-axis, you can see the percentage of biofilm formation, and on the x-axis, you see the decreasing concentrations. So we can see that the compound did show a kind of dose response as the concentration of lulwortinone decreased. But these 3 stars here are indicating that at the highest concentration, the compound was completely inhibiting the growth of the bacterium. So because of these data, we can't say very strongly that this activity is targeting specifically the inhibition of biofilm formation, as it is also obviously inhibiting the growth of the bacterium. But at least this was the activity that we managed to get out of this assay. We also did a testing for anti-proliferative activity. This was done against 3 of our in-house cell lines, a melanoma cell line, some our normal lung fibroblasts, which are included kind of as a general test for toxicity, and also against the hepatocellular carcinoma. And as you can see here, reflected by the IC50 values, so the concentrations where the cell survival is 50%, the compound affected all of the 3 cell lines. The strongest effect was against the melanoma cells with 15.5, followed by the hepatocellular carcinoma, and lastly, the normal lung fibroblasts. So some activity was seen against the human cell line. And then lastly, the compound was screened for antibacterial activity against our reference bacteria strains that we have in our lab, and also against clinical isolates that were provided by Erik Juskewitz, who is working in the research group for host microbe interactions. So I will show you now the results from where the compound had activity. As you can see here, against the reference strains, our Streptococcus agalactica and Staphylococcus aureus, it was active with MIC values, so minimal inhibitory concentrations of 12.5 and 6.25 microgram per ml. And against the clinical isolates, which are all MRSA isolates, the activity ranged from 6.25 and all the way down to 1.56 microgram per ml, which is really, really good activity. Yes. So to summarize, in 2010, Teporama isolated a fungal isolate within the Lulwortiaceae family from a dead pine in Berlevåg in the north of Norway. It was cultivated, extracted, fractionated, and tested for bioactivity, and we then saw flash fraction 5 was having some activity. A compound was isolated, and the structure was shown to be a sulfated dimeric naftopyrone. This is the first compound that has been isolated from the family Lulwortiaceae, and as far as I know, also from the Lulwortiales family order of fungi. And also the first dimeric naftopyrone, which has been substituted with the sulfate group. They also saw that this compound changed in the presence of acid. And we discovered really good antibacterial activity, some anti-proliferative activity, and also inhibition of biofilm formation was detected in our assays. So lastly, I just want to thank everyone that has been helping me with this project, especially my co-authors, of course, but also the engineers who's been helping with bioactivity testing and sequencing, and Ole Christian Hagestad, who's been helping with the phylogenetic analysis. Thank you. That's it.
Thank you very much, Marta. Have a nice and clear presentation. So I will open for questions, so please raise your hand. I hope somebody has a question, since I'm Marta's supervisor. I think I should save her for asking questions myself. So I hope someone else have a question available.
Okay.
Yes, Jean-Louis.
Yeah, thank you. Thank you, Marte, for your presentation. Is very, very interesting. I'm very curious about the possibility to work with fungi. And marine fungi looks to me very, very interesting. Do you have other strains from marine environments in your laboratory?
Yes. So one of my supervisors, Teporama, he has been working both in his PhD and after his PhD with the collection of marine fungi. So we have, I'm not exactly sure of how many, but we have quite a few. And also specifically, we have these that are strictly marine, which are the ones that I've been working with.
Okay, but it's very interesting. And do you know what is the mechanism that these fungi are able to degrade the biofilms or to inhibit the biofilm formation? Do you know if they produce some kind of enzymes which are degrading the bacteria or the biofilms, or some kind of hydrolases or glucosidases or this kind of enzymes?
So we haven't studied specifically the enzyme activity, but I know from a really, really old publication from a fungi within this family that they studied the degradation dates was on actually degradation of wood I think, because it's
Yes.
normally a wood inhabiting fungi. And there they saw like a lot of different enzymes being upregulated when it was cultivated on wood. But I can't say specifically, of course.
Okay. Well,
yeah.
Maybe some kind of cellulases then for the wood. Okay. And how do you grow them? Do you have access to fermenters in high volumes, or just doing to the lab scale?
Actually, with this one, which was something I'm very happy about, this one had a very, very high production of this compound. So normally I would have to cultivate. So we don't have big fermenters. We do culturing in flasks.
Okay.
So for some fungi, I have to cultivate up to 30, 40 liters. But from this one, it was enough with 1.4 liters, because it had a yield of 45 milligrams per liter.
Okay.
Which is insane.
Yeah.
but in a good way.
Yeah, but it's just okay for you, but it's nothing.
No, not in a,
with,
no. But in the discovery phase, it's a really good
it's very,
yield.
very.
Yes.
Okay, thank you very much.
Thank you for your questions.
Thank you for your questions. Yeah, so we have several hundred isolates of marine fungi, perhaps even higher numbers, that we are currently investigating for their antibacterial properties in particular. But so more to come, and I think also a mode of action study of this rilvotinone will be published in the next year, we hope.
Okay.
So more to follow.
So I guess you have also some isolators from seaweed biomass.
Yes.
So I think this is very interesting.
Yes.
Okay,
yes.
Thank you. I will follow you.
Great.
Thank you very much for the nice discussion and presentation. So then I will introduce the next speaker and ask her to get ready with the presentation. So it's Shaima Hassan Makboul.
Yeah.
So it's bioactive compounds.
Yes, hello everyone. Yes, hello everyone. How are you, Janet?
I'm fine, thank you. Can you share your screen?
Yeah, I am sharing now. This is my screen and let's begin.
Yes.
Please. Okay.
If you can set it in the presenter mode. Yeah, there it is. So it's just a delay. Sorry.
Yeah.
Please.
Yeah, it's okay. Yeah, please. Hello everyone. I'm Shaima Bint Makboul from Oman. A bachelor graduated student from Sultan Qaboos University, College of Agriculture and Marine Sciences, Department of Marine Sciences and Fisheries. So this is my research project in Marine Sciences and Fisheries, which is about bioactive compounds from Omani soft corals belonging to the genera Sinularia and Sarcophyton. And this project was conducted under the supervision of Professor Sergei Dobretsov, which he is the assistant professor in the Marine Science Department. So this presentation, I will have a brief introduction about soft corals in general and some isolated compounds from the corals, the aim of this study, materials and methods, then results and discussion and conclusion, then finally the acknowledgements. So let's begin. First, soft corals are actually animals. They vary in their shapes and forms and colors. They belong to the phylum Nidoria, class Anthozoa, and the subclass Octocoralia, and the order Aeracinaceae. And in this order, there are 5 families of soft corals are included, and many genera and species are included within these 5 families. So soft corals actually are translucent animals. They get their colors by the microalgae that live in their surfaces and called zooxanthellae. And they live in a symbiotic relationship with each other. And as soft corals are soft animals, they defend themselves by something called scalarities and bioactive compounds, which are physical and chemical defense mechanisms. So the idea of extracting bioactive compounds from soft corals became from them because scientists observed that these soft corals prevent themselves from the predators and pathogens by some bioactive compounds. So there are many isolated compounds from soft corals, such as marine natural products, novel compounds, and some compounds used in manufacturing like essential oils. But there is only a few studies that conducted in Oman. So the aims of this study is to test the antimicrobial activity of the soft corals, Sinularia and Sarcophyton species, and to characterize the compounds present in this species. Finally, to discuss the possible use of metabolites of this species in various fields. So here the So there is 7 species of soft corals that used in this research project. So 4 species from the genera Sarcophyton and genus Sarcophyton, and 3 species from the genus Cineraria. So here's the study scheme, starting from the collection of the soft corals, then extract preparation and evaporation, finally testing the sample by antimicrobial activity test, and doing the gas chromatography mass spectrometer analysis. Here's how the extraction of soft corals was conducted. The samples were collected from Bandar Khairan beach, Muscat in Oman, and then was coded and stored at -22 degrees Celsius. On the day of the work, the specimens were chopped, cleaned, and weighted and kept in a glass flask for extraction process. They were extracted using equal ratio of solvent mixed methanol ethyl acetate 1:1 ratio for 5 days, and this step was repeated twice for full extraction of the soft coral extracts. Then using the rotary evaporation, the crude extract was extracted using the dilute and then diluted into specific concentrations for further tests. This is the concentrations. Then the first test conducted was the antimicrobial activity test using 2 methods, agar-dust diffusion method and macro-rust method on hazardous gram-negative and gram-positive marine bacterial species. As a part of preparation step, 2 species of bacteria were cultured for 1 day, and on the day of the test, 20 microliters of 3 concentrations of 7 soft coral extracts were included on sterile 6 mm paper discs and then air dried. There were 2 extra discs for positive and negative controls, and then 3 replicates for each agar plate samples were prepared. The cultured bacteria were totally inoculated on marine agar plates with identified OD. Then the extract discs were placed on marine agar plates and incubated for 1 day. Finally, the diameters of growth inhibition zone were measured. Then the macro-rust method was conducted for 3 bacteria species I mentioned before. In this test, the metabolite activity of bacteria was observed by calculating the emitted CO2 rate in the presence of soft coral extracts using indicator solution, in which the indicator changed its color as it exposes to CO2 gas. The OD for the bacteria was identified before the treatment, and the extract concentration was 0.5 milligram per milliliter. There was a control treatment with methanol ethyl acetate, and there were 4 lubricates for each sample. Then kept in desiccator on room temperature for periods. So there were 2 readings observed after several periods. The data were analyzed by macro-rust equations, and these 3 equations, and for calculating finally the CO2 rate in microgram of carbon per milliliter per hour. Then a statistical analysis was applied by ANOVA test to compare the results. Finally, the GCMS analysis was conducted to characterize the chemical compounds in the soft coral extracts. So GCMS vials were filled with 1 milliliter of filtered soft coral extracts, and the concentration was 0.1 milligram per milliliter. There was a control vial filled with methanol ethyl acetate as a blank for the machine. So then the retention time was about 1 hour for each sample. The GCMS database was analyzed, and literature review for the chemical compounds was done using PubChem website because of the pandemic in that time, and then analyzed and compared using the Excel and PASS software. So now I will show you the result and discussion parts for the tests that were conducted, starting from the soft coral extracts itself, going through antimicrobial activity and GCMS analysis. The table here shows the weight in grams of the collected soft corals and then extracts. The highest weight of extra was recorded for the Cineraria species, while the lowest was found for the Sarcophyton species. But if we consider on the proportion of the extracted crude extra, which is the yield, we will get that this Sarcophyton species give the higher extract quantity, while the lowest quantity was for this species. Of course, this due to the variation on the fluid content of from species to species, even within the same genera. Here are the diluted and filtered soft coral extracts. As you can see, they have different color tones. And generally, the color of extracts of Sarcophyton were darker than that for the Cinderella species. Now, here's the result for the antimicrobial activity test using the agar-based diffusion method. There was no antimicrobial activity of soft corals against the gram-positive bacteria, which is Lysine-Bacillus fusiformis. But on the other hand, the soft coral extracts inhibited the growth of Pseudomonas putida species, which is gram-negative bacteria. The minimum inhibitory concentration of soft coral extracts against Pseudomonas putida are shown in this graph. The inhibition of growth was concentration dependent, with higher concentrations resulted in bigger inhibition zones. However, the diameter of inhibition zone was smaller compared to the positive control, which is gentamicin. But remember that gentamicin is pure compound, while the extras composed of many other compounds. So here, you can see in this graph, all extract inhibited the growth of Pseudomonas putida by 0.15 milligram per milliliter, while this extract of Sarcophyton species inhibited the growth at 0.5 milligram per milliliter. You can see the picture here, it is higher than even this one, this concentration, 0.8 milligram per milliliter. You know about as high concentration of cure cannot diffuse by the diffusion discs. Okay. Then the micro-RAS test was conducted in order to confirm the antimicrobial activity of the soft coral extracts. The results showed good antimicrobial activity effect on gram-negative bacteria of the genus Pseudomonas. The respiration rate was 0.25 microgram of carbon for the control. These three extracts, 1, 3, and 192, showed good resistance against the bacteria species, and the best antimicrobial resistance was by the extra Cinderella species. This graph is for the another gram-negative bacterium species belong to the Pseudomonas, with good inhibitory result as well. The respiration rate for the control was 0.28 microgram of carbon, and the best resistance was again by the Cinderella species BK115192. Here are the most dominant antibacterial compounds that were characterized by GCMS analysis in the soft coral extracts. The extract of this Sarcophyton species has those three major antibacterial agents, which showed best result in this diffusion test, while this species of Cinderella coral has this antibacterial agent, which showed a best result in micro-RAS test. Other interesting compounds were investigated and classified as antifungal, antiviral, anti-cancer, antiparasitic, antioxidant, and some compounds can be used as pharmaceutical uses and agrochemicals, and there is some compounds were unidentified yet. Then by characterizing the GCMS data, the cluster analysis resulted by 4 clusters here, 1, 2, 3, and 5, and 7, 6, 4. With similarity of lower than 15%, which means that most chemical compounds are unique from species to species of the collected corals. The second graph here showing the non-metric multidimensional scaling, in which it's showing the distinct group of extracts ordinated in 3 groups. You can see here there is some similarity between the different genera, Sarcophyton and Cineraria. This can be related to the reason of sharing of the symbionts, which are the zooxanthellae that I mentioned in the beginning of this presentation. At the end, these are the most important conclusions regarding to the presentation that I have showed you. The extracted yields of crude extracts varied from genus to genus.
Hello. Okay. I think we lost the volume, the sound. I see that there is. Can you hear me, Shema? Okay, I think we have a technical issue. I see that there's a delay. So, Shema, are you there? I think the connection was broken. So I I think that was a very nice presentation, and I would just got the conclusion. So I hope most of you have. So I think we can move on, and if she comes back, we can perhaps have some questions for her. Maybe I give her a few seconds to see if she can log in again. Okay, but then I think we move on to the last speaker, and hopefully she will come back. So then I will invite Hafid Al-Marouki from Malaga University to perhaps share the screen and continue with the presentation.
Hello, everybody.
Hello.
I'm connected. Can you hear me?
I can hear you well, so
Okay.
if you can share the screen.
Here you are.
Yes. We see it. So yeah, so now it's in presenter mode. Thank you. Please.
Hi, everybody. Welcome to this very fast presentation. I'm Hafid, originally from Oman as well, like Shaima, but I'm doing my PhD in Malaga University in Spain. That's like small connection. What about this presentation? I just need to. So, brief about I need to just minimize this. Sorry, I'm not so good with the Zoom. Okay. This presentation is about the effect of fertilizer concentration on productivity and biochemical composition of spirulina using urea as a nitrogen source. Brief about why we care about this study, because in Oman we have, I think it's also internationally, there were over extraction of underground water, and there is water scarcity everywhere, almost in the agricultural areas, and also inefficient overuse of the extracted water and less fertilized land. And it's one of the major reasons for the food scarcity, actually. And this caused, of course, the seawater intrusion, and people also lose them farm, which is main source of them income in Oman, for example, and I think also the same internationally. And in Oman also they start to long time trying to solve this problem by making new rules regarding using the agricultural land and water and constructing dams and tolerant crops. However, there is a limitation with this. Consider the time and also very expensive and not so much efficient. That's why farming spirulina and microalgae is one of the solution for such a problem. And many studies actually has been showing that microalgae is like one of the future coming for food, and spirulina cultivation is like perfect for the arid area in this desert zone like Oman, and in Middle East it's like the only country actually developed such commercially is only Israel, and hopefully we are doing the same soon, because in Oman, for example, we have like almost zero farm for microalgae. And in the north of Oman, where almost 70% of the vegetation, now where the seawater intrusion, it looked like how in the photo, especially in Al Batina zone in the north, and most of it because of the overpumping of the water. So in this study, we actually investigate the effect of urea as a nitrogen source, and also we diluted the standard medium to 50% and 25% to investigate like the effect of that, and also to determine the biochemical composition of two strains that we used, one from Spain, Malaga, and the second from Malaysia. And brief about microalgae as, for example, biofuel. It's not economically viable, especially in country like Oman, which is like oil producer country. So it's not commercially applicable. So that's why spirulina could be like very intelligent idea for food and feed. And currently, there is no actually so much research regarding the cultivation of microalgae in that area. And most of the experiments, it's like just lab-wise only. Why spirulina? Because it's has less risk of contamination, and it's considered as a superfood. Very high protein content, up to 60 to 70%, and it survives in very harsh conditions. Very high antioxidants, essential fatty acid, and it's very easy to be cultivated as in mass since very long time already. And actually, one of the main challenges for spirulina cultivation is the cost. 50% of the cost of spirulina comes from the fertilizer. And many studies has been already conducted using the NPK fertilizer. NPK fertilizer is the most common agricultural fertilizer. And also using the waste of fluent or organic waste, palm oil waste, and also farms waste. To all of them is like to reduce the cost of the fertilizer. In this experiment, we have like this is the flow chart of the experiment. We have like two strains from, as I said, from Malaysia and Spain, and we diluted the standard to 25 and 50%. And we check the growth performance. We harvest and freeze dry them for the chemical analysis. We check the protein, carbohydrate, lipid, chlorophyll, and cyanine. And then we through ANOVA test, of course. And regarding the medium, so a lot of numbers, which is of course not very interesting for you guys, but I will focus only to the main, which is the bicarbonate, the sodium bicarbonate, which is considered as almost 70% of the fertilizer. So we can see in the standard Zaruq medium and both, it's like 16 grams per liter. It's like almost 75% of the total fertilizer. And the second one is the nitrate, which is almost 20%. In the standard Zaruq medium, there is nitrate, but in the modified, we used zero nitrate, but we used urea. So we cultivate the spirulina indoor to adapt for the new medium. Then we move to outdoor to adapt to the high intensity of light. In Oman, the experiment was. And then we prepared the medium and we cultivated it inside the greenhouse. So in 10 liter in triplicate. After that, we harvest, we freeze dried, and we continue the analysis actually in Malaga University for the biochemical analysis. So in the growth performance, there were no significant effect between the strain and no even between the 50% and 100% up to day 7. So on day 7 to day 8, the 25% have like spiked down, but up to the day 7 it was fine, and it was not significant. It was significant effect like compared to the 25% was significantly different. In terms of the productivity, the same. 50% and 100% show the same result. Only 25% was less. In terms of the protein, and I believe if we harvest on day 7, it will be the same, but it got stressed on the day between day 7 to the day 8 before harvesting. And in the protein, 50% and 100% fertilizer show the same result. 25% was significantly less. And carbohydrate, because spirulina, when it gets stressed, it converts, it produces more carbs. So 25% was significantly different, like high in carbohydrate compared to 50% and 100%, which show the same content. Lipids, the same. They were also not significant difference between 50% and 100%. In 25%, they were like significantly different compared, but not in all the treatments. Also, the urea one showed a significant difference, but the nitrate when the one with nitrate was not really significant. In chlorophyll and phycocyanin, also the one just that in circle, the 25% with the urea was significant, but with the nitrate was not, except the phycocyanin from the standard Zaruch medium. So in conclusion, this study showed that there is no significance in between the Spanish and Malaysian strain within the same fertilizer. And also there is no significance within the same group, but 50% and 100% show like no significant at all. 25% was not significant up to day 6. Then because of very low fertilizer, it was like start to decline. And why we choose urea as a nitrogen source compared to sodium nitrate? First of all, urea is a very cheap source of nitrogen. Also, we used only less than half gram per liter compared to the standard of the nitrate, which is 2.5 gram per liter. And in total, it's like 82.3% less in quantity. So the suggestion of the production of spirulina in terms of cost can be significantly reduced up to 75% in terms of the fertilizer. And within the new technology in the harvesting system and mechanized technology of production of spirulina, so the cost of the fertilizer can be increased up to 70%. Not only like until now, the cost of the production of spirulina is 50% of the total cost. But when the technology is increased in terms of the mechanized and other things, so the price of the cost of the fertilizer also will increase. And by that, if we manage to reduce the cost of the fertilizer, we will decrease the total cost of production of spirulina. And Oman is one of the best, I mean, the desert is zone, the arid area, one of the best weather condition to grow this type of microalgae. That's why this type of study, we really need this type of applied science for this area of the world. And that's very fast. I think I tried to do it like very fast. Thank you for your presentation. I want also to mention that we luckily published already this paper in one international journal. And thank you for your attention, and welcome for your questions.
Thank you very much for this presentation, and also not only emphasizing the technology, but also the bioeconomy or economy of this. I think this is very important to include. So I will open the screen or floor for questions. So please raise your hand. Or you can just unmute if you don't find the raise hand button. Okay, there, Marta.
Hey, thank you for the presentation. It was really nice. I have to say I don't know very much about this, so the question is reflecting that. How do you eat this? Is it like how is this kind of consumed?
Okay, so it's basically spirulina has many shapes like to consume. Can be in fresh paste and can be additive to juice. Like this is in the market. I'm speaking about market size now. Market wise. I saw it in Germany, for example, as kiwi, spirulina kiwi, and there is also there is a lot of bread with spirulina. I saw also in Portugal in Faro, in Portugal, South Portugal. One day I was there, and for the breakfast I ordered also. There is a banana spirulina nuts, like a dish of for breakfast. This is like as in the market also, but like home wise, you find it as belts, as pills, additives, supplements. Or powder, which is more recommended because when it's like powder shape, it's very extremely cheaper. However, in total, it's still expensive. So here I'm in Malaga, I'm buying it from the market. It's 200 grams in the market with 16 euro for only 200 grams. But when you go to quantity, in Oman, I bought it from the Carrefour, half 450 grams with like 10 euro. But if you compare it with the like the food in general, this is a very expensive product. And then at home, it depends to you. Like you can make it like shake. Generally, I make it in my shake before going to the gym. It doesn't taste like good, but you get used. If you like seafood, it's almost like seafood.
Yeah, that was my next question. If it tastes good, but then you answered. So.
Actually, one more thing is like if you type on YouTube also, like as a cosmetic, it does use it as face mask. It's not only actually for food. It also can be like for cosmetic. If you just type spirulina mask, you will see like a lot of videos also.
Okay, thank you.
Thank you very much for your question. We have time for another one if there are a question. No, okay. So thank you again, Hafid, for this really nice presentation. I can then ask you to stop sharing. And
thanks, guys, for the organizer, especially.
Yes, so thank you. And I know now that Shaima has arrived back, I hope. So if, are you here, Shaima?
Yes, hello,
Yes.
I'm here. I don't know how I disconnected with you. I don't remember in which slide we have.
You were on the conclusion, so,
really?
so you were almost at the end.
Oh my God, Alhamdulillah in Arabic, yani.
Yeah. So then I would just, if there are anybody in the audience that have a question for you, we might just take one or two questions. So, so if people,
I have, but I want to share my screen if you don't mind. Yeah. Yes, here. Sorry for interrupting. I don't know what's happened to Wi-Fi, so I connected by my cellular data now.
Yeah. So if you can just take a minute to summarize your talk.
This summarize. Yeah. Is then the best antimicrobial activity was for this extract, saprophyton extract, by this diffusion method, and the best antibacterial activity was by this cinereal soft coral, which done by the microrasp method. And then the GCMS analysis showed interesting bioactive compounds and unidentified compounds. So my appreciations and hard thanks to this team of professors and doctors and misters that worked in this project, and also for the people that behind the scene. And then special appreciation for Professor Sergi, who is the project supervisor in this project. And yes, thank you. If you have any questions. Yes.
Thank you very much. Then we got all of your talk. So I don't know, Jean-Louis, you have raised your hand. Is it for Shaima? You have a question, but please.
Yeah, yes.
Yeah, please.
Yeah, thank you, Saima, very much. I like very much your presentation. Just a easy question. Have you checked the possibility to use material from cultivated origin? Does these corals can be cultivated somewhere?
No, actually, if you know, this corals still didn't identify it genetically. We are working on it, so we can publish the paper. So in Oman, we don't have many studies on soft corals, also in fungi and marine plants. So yes, we don't know anything about these soft corals. Even we are just discovering it and studying it.
Okay.
Yeah.
So
But,
go ahead.
yeah,
It's very interesting.
Yeah, if I have any idea, I will tell you, of course.
Okay. Thank you.
Thank you.
Thank you very much, Shaima, for coming back to us after the
I'm
internet.
sorry, I really, my apologies.
No problem.
So
I think we're almost there. So
Yeah.
thank you very much. And
Did I stop sharing now?
yeah, you can stop sharing. So I would just say thank you to all the speakers for a really, really nice program. And I see Maria is here, so I will leave the word for Maria, please.
Thank you very much, Jeanette, for co-chairing this session, and thanks for the amazing talks to everyone and for joining us. I'm just quickly going to share again the presentation, just with some links potentially. As I said, I'm gonna stop recording, and thank you again. Stop recording, and oh, sorry, I just stopped sharing actually. So now I'm going to stop recording.